All right, let's get started. Pardon me. Let me get rid of that extra element here on the screen. Okay, yeah.
I have a question. Many projects, when you do that, as long as, like, let's say we answered the questions and we showed that we had mutation and recombination in our algorithm, and we converge to the right solution, is there anything else we'd be looking for kind of the grading?
Well, there is a rubric, so you can, if you go to the assignment down at the on the side or down the bottom, if you're in Canvas, you can click on rubric, and then you can see the things. But the biggest thing is making sure to answer like for question 1, there's like 3 bullet points, and to hit all of those. For question 2, there's sort of a main, like sort of you have to show some, I think, a graph and make some comments on comparison. And then for question 3, the bonus one, kind of a similar thing. So the big things, as you'll see in the rubric, are kind of making sure you hit those. And then there's maybe a couple of other small things about, inclusion of, make sure you've included code, things like that. You'll see like those are maybe a small part of the points, but there is a rubric there at the bottom, which, and then plus in the assignment, I think like for the first bullet, I say it's 5 points, the second bullet's 5 points, and so on. So yeah, any other administrative type of comments? I might turn on a little more light up front here. All right, well, that's about this for you. Okay, so any questions or comments online? Everybody looks about good. Okay. All right, so we are now talking about the immune system and immunocomputing and artificial immune systems. And like I mentioned before, immunocomputing, it's sort of the broader scope here, is when you look to the immune system for inspiration for algorithmic design. So the immune system, as we'll talk briefly about in this unit, does a lot of information processing. It has memory, there's storage of that memory, it has to manage that storage of the memory, has to be able to recognize self and non-self. It's very flexible, it's plastic, it changes over time. There's a lot of really cool things you can learn or get inspired by looking at the immune system, and that falls under this idea of immunocomputing. And we are focusing under a smaller set of that, these so-called artificial immune systems. Which we'll also call AIS. And as we'll talk about in just a second, I'm mainly going to be focusing on negative selection and clonal selection algorithms. And we will define what those mean in a moment here. And so the immune system in general, when we think about at least the immune system in vertebrates, I mentioned last time when I was going over and not realizing it, that that the immune system in vertebrates is broken up into 2 major systems: the innate or non-specific immune system. Waiting for the pencil here to come back. It usually takes a second. There it goes. And the innate immune system, this is very ancient. So this is inherited from our relatives in the phylogenetic tree that go way, 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 way back. And so you find the innate immune system back in fungi and plants. It goes, it's kind of the default immune system that you have for most life on this planet. And the innate immune system, it's generic, which means it is primarily meant to identify generic invaders or intrusions and mount a non-specific response. And so we often refer to these responses as an inflammatory response. And so if you've ever, whenever you get, a spot that, turns red, it maybe gets warm, so you have increased blood flow. This is an inflammatory response. And so the idea here is your body has recognized that there's something wrong, something foreign. And so what it does is it starts sending a bunch of resources into that area. It heats up that particular area. It brings blood flow, it brings some other things into there, and it brings parts of the immune system that are pretty good at recognizing self, and they also recognize when self is damaged. And so there are aspects of your own cells that either due to infection or due to cancer start presenting slightly different, bumps and lumps than they would normally, and the non-specific sort of general system is able to find them and take them out. And so that's, it doesn't learn anything. It's sort of just, for the most part, people are learning more about the innate immune system, and it does have maybe some ability to learn, but for the most part, it's just a generic response to to your body taking in something that's not supposed to be there. And so if you get like your first vaccine in a series, like, say the COVID vaccine, then, that pain that you get around the injection site, the low-grade fever you get, those are all actually your innate immune system saying, this is different, it's maybe not supposed to be here. And so it starts heating up, it starts directing resources into that area, and that's your body sort of responding to a generic threat. Now, in more specific to vertebrates, there's also the acquired or specific, what are some other names we give to the, or an adaptive immune system. And this is one where at least the computer science likes to focus on, because it does have memory, it changes over time and so on. So in the adaptive immune system, it is where you have immunological memory, which means that it can actually remember specific infections. And so you can get infected with something, it will recognize things that it's never seen before. It will actually create elements in your bloodstream that are tailored to those specific things, and it'll keep them around for a certain amount of time. So it actually is able to build up memory there. And so it is highly specific. And so, and one of the things that the innate immune system does is it actually recruits the adaptive immune system. And so there is recruitment So on your second or third, your booster shot in the vaccine series, a lot of times you have a very different response. And some part of that is actually when that now your body has actually recognized this thing before, and it mobilizes a specific response. And that's why you get a qualitatively different feel. So this is, so this, again, it's highly specific, but it does get recruited. So if your body notices that there is some weird thing that's not supposed to be there, along with bringing the general purpose defenses, heating up the area or maybe your whole body or whatever, then it also can actually recruit elements of the adaptive immune system to check it out and say, have you seen this before? Do we have more targeted defenses? And then it'll mobilize those targeted defenses. And so how does it do this search? So the big question here is, I'll go back to my purple color, is how does the adaptive immune system generate variation? I'll say generate and shape variation to build specific responses. And it turns out that a lot of the mechanisms that are the fundamental mechanisms that go into this in biology actually come from co-opting mechanisms of genetics. And so there, as I'll talk about here in a second, you see that some of the same things that generate and shape allelic distributions in populations also shape the distributions of different types of highly tailored responses in the immune system. And so that's, and so how does it do that? So we know that at least in populations, natural populations, you get variation through a number of different mechanisms. You can get mutations, but they're pretty rare. Like how you get a mutation due to like radiation or cosmic rays, whatever, that doesn't happen that often. Or you can get, recombination. You can sample from two different individuals to create a new individual. It's a different mix. That's kind of a thing that could happen a lot. Or you have mistakes that are made. So when DNA, I've heard DNA is a double helix. It gets split off in two, and then there's this template that gets copied, and then it gets put back together. Well, the copy that's made, sometimes it's a bad copy, and that generates these mistakes. And that's another way you can get these sorts of things. So those are origins of variation in like kind of populations. What the adaptive immune system does, and I'm not going to go into much detail at all about this, but I want to at least mention it because it comes up. What the adaptive immune system employs is something called VDJ recombination. And from a computer science perspective, it's pretty cool. So, so VDJ, this stands for variable diversity joining, right? So this is a variable. I'll just write this down this way. Maybe I'll just, for space, I'll say variable. And this isn't something that I care that you know off the, that you should memorize or something like that, but it's something that I think is good to have exposure to. And if you're, and those of you interested can look up more info about this. And so what this VDJ, I'll highlight that, variable diversity joining. So recombination, this is, we've heard of recombination before. This normally only happens during sex, where you've got two gametes, so you've got a sperm and an egg, they come together, you got 23 chromosomes in one, 23 chromosomes in another. They need to assort and pair off together, but you end up getting all of the, for each chromosome from the biological father, you've got the chromosome from the biological mother, they come together, and instead of just joining together like that, they twist and recombine so that the new chromosomes that are made are going to be mixtures of the mother and the father in novel sort of novel recombinations there. And so it's combining from these two. Now, in most of your somatic cells, so not sex cells, like just, you're growing and things like that, you leave the DNA alone. You don't do this recombination stuff. You just sort of, you got nuclear DNA in a cell, you produce a new cell, it's got the same DNA as the old cell. Except in special cells in the adaptive immune system uses its own version of recombination to recombine from a bunch of elements to create a huge combinatorial explosion of possibilities that it can generate. And so inside these special cells in the adaptive immune system, this is also referred to as somatic hypermutation. And so somatic just means it's a body cell, it's not a sex cell. And hypermutation means way more variation than you would expect to get through normal other sort of mechanisms there. And so in the normal process, and this is sort of, I guess I'll put it this way, nature's random number generator. And so without getting into too many details, there's basically there's a region across a couple of different chromosomes that in each region has a bunch of these V variable sections, a bunch of these diversity sections, and a bunch of these joining sections. And when you create new cells that can recognize what we're going to refer to as antigens, the antibodies that they create are built up from random selections from all the variable regions, the diversity regions, and the joining regions. So it can look at this sort of, it has like kind of a kitchen full of ingredients, and it can then choose random bits of these things and recombine them in creating sort of these just all sorts of these new alleles. It can kind of just like a roulette wheel just come out and get these random outcomes of this huge number of alleles, like something like 10 to the 11th or something like that. It's a huge number that it can sample from. And each one of these things generates an antibody with different abilities to match antigens that come out from the outer environment. And so this is just a way that it can generate a huge amount of diversity. And like I said, it's just nature's random number generator. It's a beautiful system to generate variation. So this is how variation is generated. But then we say, well, once we have this variation, we have to solve these two problems. One, how do we prevent attacking ourselves? And so this is self versus non-self. So this is preventing an autoimmune response. And so this is, I'll call autoimmune regulation. And then the other problem, or the other thing we need to solve is once we, how do we enhance antibodies that have matched to foreign antigens? So those are the two problems that we're going to focus on here. I've generated a whole bunch of antibodies, and these antibodies, if they match something in the bloodstream, they're going to mobilize a specific response. They're going to attack that thing that they're matched. And so I want to, one, make sure they don't attack normal stuff. And then two, if they find something, then I might want to then increase the number of the population of those, because now it's not just I found a random antibody, I found an antibody that's actually useful. And so this first part here motivates what's referred to as negative selection. And this second part is going to refer to algorithms that we talk about called clonal selection. And what we'll see is that clonal selection is going to look a lot like a genetic algorithm without recombination. So we'll see that in just a second. Now, I should mention that negative selection here, this was largely kind of discovered and popularized by a woman named Stephanie Forrest. and her collaborators. And I mentioned Stephanie Forrest's name because she is a distinguished faculty member in the School of Computing and Augmented Intelligence. And so she, a couple of years ago, came to ASU and started a center for biocomputation, society, and security. She does a lot of cybersecurity work. And a lot of her career started with looking at negative selection algorithms, so artificial immune system algorithms. And she still does work that are inspired by the immune system to catch threats and anomalies and things like that. So, so it's kind of a local thing there. All right, so that's the basic overview of at least the biological motivations. So before we then go into the sort of specifics of how negative selection and clonal selection work, then are there any questions about this basic structure here? So I'm not going into a great deal of detail about, I mean, you could go, the immune system it's an extremely beautiful, elegant, and deep system. Both of these systems here, there's a lot of cool things you could talk about, but I just want to put these up as kind of this motivation that the specific immune system is sort of the motivation for these two things, and the basic ideas here of what the problems the specific immune system has had to solve have almost been more influential than the precise mechanisms of exactly what the body does to solve those in its particular way. All right, I see a question online. So VDJ generates a bunch of antibodies. Negative selection takes out those that may attack us. That's a decent way to summarize it. Is that VDJ is the mechanism used to generate diversity in antibodies. Some of those antibodies would attack self, and so I'll get to that here in a second. So this for negative selection, the background here is that we have. So and this is in part to answer this question. There is an organ called the thymus, right around here. And when all of these antibodies are created, the first thing they do is they get shuttled into the thymus for processing. And so they go into the thymus, and the thymus has got a cool system where it's got a bunch of probes, where it says, here's a bunch of samples of things that are pretty diverse, that are all examples of self. These are all things that are specific to us, me as an individual. And so, so if you happen to match against these, that's bad, and we're going to get rid of you. So the thymus challenges their antibodies with examples of self, and then it removes antibodies that match these examples. And it flushes them out. And those that can manage to pass through, it's kind of like a filter or a sieve, where it catches all those ones that match to self. Those are the ones that then go out into the bloodstream and then start looking for new stuff. And so this isn't perfect. And so there are peripheral mechanisms that are away from the thymus that continue to tune these, but most of them get caught in the thymus and get stripped out and then discarded. And so that's where negative selection is inspired by, this process in the thymus of presenting exemplars of self and getting rid of anything that matches them, and then deploying anything that's left over. So, so I'm not, I mean, it's a really simple idea. And so, whereas in clonal selection, I'll get the question in just a second. In clonal selection, I'm going to give a full algorithm in great detail on how clonal selection works. It's going to look a lot like a GA. But this basic idea of negative selection can be implemented in a bunch of different ways. But the basic idea is exactly that. Generate a bunch of random patterns or random mechanisms for matching patterns, and then and they're basically classifiers. They're sort of like a one class classifier. It's either in this class or it's not. It's either self or not. And anything that matches self, you get rid of. And then anything else you use as a possible classifier, because if it doesn't match self, it's okay if it doesn't match anything else. If it happens to match something, but it didn't match self, it's probably foreign. And so then we're going to end up using it as a classifier of something that's wrong. Yeah, this question?
So could you say that we could have used negative selection in our genetic algorithm for the mini project, because when we were like, if the solution didn't fit the range of like -0.5 to 1, we kicked those out. So could that been a form of negative selection that you implemented?
So there was a question of, can we interpret constraint in the genetic algorithm? Can we interpret the throwing out of solutions that fall outside of our feasible region as a form of negative selection? And I guess you could consider that as sort of a little negative selection step. I think we usually don't call it that because just it's not inspired by that. But on the other hand, this idea of this feasible region is kind of an artificial, like it doesn't come from biology anyway. So yeah, I mean, you could, if it helps, you definitely could call that a negative selection. It still ends up kind of mixing a metaphor, which is, I think, the reason why people probably maybe don't go that route. But yeah, absolutely, that would be a simple version of negative selection. So if you look in this unit, you'll find several readings on negative selection. And a lot of them, I think I've included, will include Dasgupta and also Forrest. And I don't want to survey all of these in detail, but I encourage you to kind of at least look through some of the titles. And it'll give you an idea of applications where this has applied. So as an example, I think in the upcoming, so there is a concept check, auto-graded concept check on genetic algorithms due Sunday. Then the concept check on this unit includes, I think it asks you to do a reading on these, on one of, I think, these Dasgupta papers, where I ask you about this machine shop example. I think I chose the machine shop one, where what they did is they said, well, what we're going to do is have a sensor which is able to read acoustic measurements from a tool as it operates normally. And so this tool might be a drill, for example. And so as the drill's operating, it can pick up acoustic measurements from this drill. And you can then use a bunch of different signal. You could just imagine, well, there are features of the sound of a drill that I could possibly match. And so randomly, you could just decide, I've got a strategy where I'm going to look for certain frequencies, for example. So you could say, for example, that I am going to run the sound through some Fourier transform, and I'm going to look for certain frequencies. Now, I don't know which frequencies to look for, but I'm always going to look for 5 frequencies. And then you could randomize which 5 frequencies you look for. And so you have sort of a battery of possible 5 frequency tests that you could apply to this acoustic signal. So you generate all of these tests, and then you run the drill under normal conditions, and then you see how many of those tests are hit. And all of the ones that end up being triggered by the drill under normal conditions, you throw out. And then the rest of them that are left over, these are 5 frequency combinations that didn't get hit under normal operations. And so then you just deploy this thing, and it's constantly listening for these things that it should never hear. And if it does hear them, it raises a flag and tells somebody that maybe the drill is about to fail, and you should go and investigate it. So the key point of negative selection is not training the classifier on particular failures. It's training the classifier on the normal operation, generating a random set of classifiers, throwing out the classifiers that detect normal, and just leaving in the rest of the classifiers that didn't detect anything in a normal operation, with the hope that there might be classifiers in there that will detect a bunch of different anomalies that you have not anticipated could happen. So if you know the type of anomaly you're looking for, this isn't the tool that you would use. So negative selection is, is typically used for, I'll just mention that down, anomaly detection. And you probably could draw a connection with the so-called literature on one-class classification. So if you look up one-class classification, you can find it. But where the point here is that you do not know anomaly ahead of time. So you generate a bunch of classifiers, throw out the ones that recognize the thing that is good, hope that you've got a classifier to something that's bad left over. That's one negative, yeah.
So I can understand, like, when does the virus start to make new classifications? Because if it's constantly just adding foreign elements and it's starting to self-elements, I guess, when does it start to make other classifications and when does it stop? Like, is there a limit to that?
So there's a question of I think, when is the variation being generated in the immune system? And it's in some ways, really the VDJ combination. So that's a much deeper conversation about how the variation is regulated in the immune system. The simple answer is that in your bone marrow, for example, so ultimately we're talking about white blood cells. Blood cells are generated in bone marrow and some other regions of the body. And when white blood cells are created, this VDJ recombination is kind of always going on. And so there's always new variation being deployed, and then it then gets shuttled up to the thymus, and then that's where it gets filtered there. So the thymus always has kind of one template for self, and just through the process of generating new white blood cells, this is just a standard scrubbing process that goes on as these new things get generated. So there are things that get scrubbed out that are going to get regenerated in theory, but there's so much variation. Again, it's like 10 to the 11th or something like that, that it's probably unlikely for a lot of the things that have been scrubbed out to get rediscovered in high numbers. So it's just kind of constantly going on. Yeah, and so autoimmune disorders are where your body attacks itself. That's really troublesome because basically it means that your body has figured out how to make something that it doesn't know how to filter out. So that's why people who have autoimmune disorders often need to get something called an intravenous immunoglobulin transfer, which means you actually take your blood and you run it through a machine, and it scrubs out all of these white blood cells and replaces them with white blood cells from someone else, so that they can be filtered by your own non-self processes, but they don't have the deleterious one that's not getting caught. So yeah, autoimmune disorders are really tricky because of that. But the natural sort of nominal behavior is that autoimmune regulation is just constantly going on in the thymus. So I hope does that answer your question? All right. So I don't want to go into much more detail about negative selection. I just want to marinate you with the basic idea. It can be applied in lots of different ways. All you really need is a way to have sort of a randomizable classifier. So this is sort of the key here, is that you need a sort of I'll say a randomizable classifier and samples of self to filter the ensemble of classifiers. So there's still kind of population intelligence going on here, where we're not building a single, it's just like with the genetic algorithm, we're not building a single strategy, we're building a huge number of strategies, and that's going to be limited by our resources. Just like in the genetic algorithm, it's limited by our max population size. And then we filter that group out so that we put holes in it where the things that we do not want to account as anomalies, and then everything else becomes an anomaly detector, which we may never use because they may be coding for anomalies that are impossible. And that's negative selection. So any questions on that? See online, I see there's a couple of questions, or at least one. Could you go over an example when the body would attack itself? So that's, and there's a bunch of cases. Allergies are one kind of example where your body will recognize allergens that it probably should just let go, and it makes you feel like you're sick because it's trying to attack something that actually is benign. Other examples, like I mentioned IVIG. The reason I know about IVIG is my father has a condition where his immune system started to attack the nerves in his back, and it would give him this banding and give him a paralysis in his legs until he got IVIG to flush those things out. And he was able to reset his immune system and stop this process. But it was this runaway process where where his own immune system found a combination that ended up getting amplified, would actually attack his own nerves and attack the myelin sheaths around the nerves, so that his nerves would short circuit and create this paralysis. And he still, because he had to go so long with this paralysis, he had sort of muscle damage and a little bit of nerve damage in his feet, so he can't kind of straighten his feet out very much anymore. But since they stopped that process by flushing all those out, bringing new ones in, and kind of resetting his immune system, then he hasn't had the problem since then, knock on wood. He's always worried, though, whenever he gets a vaccine or something like that, that he's going to sort of energize his immune system to possibly get back into this region where it amplifies these bad cells that attack his own nerves again. So he's always sort of very paranoid whenever he gets anything that feels like he's, numbness or anything like that. He's afraid that it's going to come back up. So there's tons of examples where you get these. Some of them are autoimmune disorders, like HIV, and AIDS. That's an example of an autoimmune disorder, but that's caused by a virus. But there's a bunch of other cases where people have a natural immunity to themselves. It can be induced also by organ transplants. And so you get a new liver, you have to take immune suppressing drugs because that new liver does not have the self markers that your thymus has. And so your white blood cells that are created actually will attack this organ because even though it's a perfectly good organ doing the things all good livers should do, it still does not look like your liver, and so it ends up getting attacked. And so they're experimenting with now organ transplants from genetically altered non-human animals that are less likely to express the proteins that would generate this autoimmune response, so you don't have to take as many of the immune suppressing drugs. That's another reason why people who have had organ transplants can't get a lot of the vaccines, like the vaccine for COVID, because they're on immunosuppressing drugs that actually prevent the process that would normally happen to develop that specific immunity, because their immune system has been so suppressed that the vaccine goes in and it just gets flushed out. So tons of examples, and if you're interested in those, just look up autoimmune disorders, autoimmune reaction, just the phrase autoimmune, AUTO, immune, self-immune, and I think you'll find a bunch of different literature on that, or at least lists of things that fall into that category. All right, I think it's that. Okay, so that's negative selection. It's kind of a cool thing. It's just because it's so simple, it's been applied to cybersecurity, it's been applied to industrial anomaly detection, a bunch of different things, just a really simple idea. Now there's something that's a little more specific than that, no pun intended, is the other kind of big branch of artificial immune systems are clonal selection algorithms. In which, whereas with negative selection, I give you kind of a high level view, in clonal selection, I'm just going to write out a sample of a clonal selection algorithm, and then so we can build up the discussion from that. So clonal selection is similar to a GA without recombination. And so the idea behind it is we start the algorithm up with an input set, I'll call it S, of antigens. I'm going to call them antigens, but of course this is, this is an algorithm, so we're just calling them that. These are analogous to things that would be on foreign bodies like viruses, for example, that could be matched by the immune system. So these are, so it's kind of like a negative selection. We provided samples of self. Here we're providing samples of non-self. So the antigens are the things we want to match, but we don't know the best way to match them. So we need to create a set of memory cells that are best matches for these things. And so what I mean by that is this could be abstractly, you could say, my antigens are particular attacks to a system. So I know that I could have SQL injection attacks, for example, on web systems. So you can come up with all these different cybersecurity attacks. And you don't know the best way to detect these attacks. So it's not so simple. Like when I talk about antigen, it's something I'm talking about a lock and a key, where I'm like, well, if I draw an antigen, I just I draw shapes and I just find the geometric shape that fits into that. I'm done. I don't need an algorithm for that. But that's not kind of what I'm saying right here. I'm talking about antigens. You can think of these as sample or types of attack to be detected. And my goal is to generate an output set M of, I'm going to call them memory B cells, which are ways to detect these attacks. So SQL injection attacks might be hard to detect because there's a bunch of SQL traffic that's going around the network anyway. So there's so how do we detect these particular, and we don't know exactly what the attack's going to be. We just know that they're going to co-opt our web infrastructure to then insert things in our database. So these might be SQL commands that we not normally expect through our web traffic, but they're going to look pretty close to that. So we don't know exactly what these commands are, but we need some way to sort of detect them. So we have a sample of the types of attacks, and we need ways to detect those attacks. So how do we do that? So the algorithm, it's going to start by generating a random B cells. And so these are random detection mechanisms here. And then for all, or I guess I could say for any, but for all antigens, and I'll call this particular antigen in this loop AG, but in my set of antigens. So for every type of attack, we do the following. We are going to calculate affinity between, I'll say affinity of all B cells to the antigen AG. And so what do I mean by calculate affinity? If I have a particular type of attack, then I'm going to see, for example, how likely was the particular B cell able to detect this particular attack? So did it detect the attack or not? And if it's a classifier that outputs a number between 0 and 1, and or if it's if I need to threshold it, so before I threshold to actually generate the classification, you could just think of this as, well, this is the actual, if this is a regression output or something like that, this is like the probability that this is an attack. And so how many of these B cells generate high probability numbers versus low probability numbers. So this is a tool that generates a high number if it matches, a low number if it doesn't, and anything in between if it's not quite sure. So then we select, and you can think of this, if it helps, as kind of a fitness objective or calculation of fitness in the GA sense. So then we're going to select the highest affinity B cells. And we are going to, once we've selected the highest affinity B cells, we will do proportional cloning, just like proportional selection. And so the idea here is that we've got a bunch of B cells, let's say we've got 100 B cells, we're gonna choose the top 30 of them, and then of those top 30, we're gonna look at their affinities, like fitnesses, and then based on their affinities, then we're going to clone each one of them, so that the one with the highest affinity will get cloned the most, the one with the next highest is further down. So it's just like fitness proportionate selection or ranking selection. So there's a bunch of different selection operators that you could imagine going on here. So I could call this process here like a selection operator. And then there's no mutation or recombination. And then we take those and we place clones in another temporary set calling C. And then here's where we're going to generate new variation, because then we've taken all our clones and now we're going to put them, and it's like our offspring. So you can view these maybe as offspring before mutation. And then for each one of these clones in C, so I'll say for all clones in C, then we are going to mutate it with a rate that is inversely proportional to their affinity. So if they're already a really good match, we don't mutate them. If they're a really poor match, we mutate them with a high rate. And so that helps us keep sort of it's like a version of elitism where it keeps the high performing ones in the population and it flushes the lower performing ones out. So mutate each at rate inversely. I'll maybe write that out. Inversely proportional to affinity. And so there's our mutation step here. And then from there, we can determine new affinities after the mutation. It's like determining the new fitnesses. And then we can take all those clones and we can copy them into B. And then we're going to copy the highest affinity clones, AIS, into M. And we replace the lowest affinity B cells with randomly generated alternatives. So replace lowest affinity B cells in B with random alternatives. And so we could imagine that, so this here is our new population for the next iteration. And then these here are our output solutions. And then we can repeat up to here. And that's basically clonal selection. And you can imagine that going on continuously. So you could imagine this terminating. So, maybe termination condition. Or continuous operation, where you might even change the antigens over time. So some things that I kind of want to point out here is that, like I said, it's like a GA without crossover. It's a little bit like, we haven't talked about multi-objective algorithms yet, multi-objective genetic algorithms yet, but it has a nature that's similar to a multi-objective optimization problem because we have multiple antigens that we're trying to track. You can think of the affinity towards the antigen. It's like a minimization problem or a maximization. I want to maximize the affinity, but I want to maximize the affinity simultaneously to every one of these antigens. So it might be that a B cell that has high affinity to one antigen is a really poor match for another antigen, but I need B cells for both in my population. So this is attempting to create B cells that match all sorts of antigens simultaneously and keep them in there. And so even though sometimes I get B cells that have poor matches, hopefully if they had a good match to another antigen, they exploded in the population for that antigen so that there's still enough of them sticking around in the population when they got challenged by this antigen. So this is the idea of clonal selection. So cloning things that do really well, mutating the things that don't do very well, and hopefully establishing a population that match multiple challenges simultaneously. So I'll just put that in here, the different color here. We're trying to match multiple challenges simultaneously. All right, any questions about the basics behind clonal selection? Yeah. Affinity is abstractly, it is how well an antibody sticks to an antigen. In the concrete case, it is how well does your automation thing match the challenge that you put in. So if I put an SQL injection attack in, I put that as a sample, then I've got some classifier, a neural network, whatever, that I put that thing in and it spits out a number between 0 and 1, where 1, it thinks it's definitely an attack, 0, it thinks it's not. Affinity would be that 0 to 1 scale. So if it comes up as a 0.9, then that means that, oh, I've detected the SQL injection attack, so it's got high affinity to that particular antigen, the SQL injection antigen. Comes up as a 0.1, then it doesn't think it's an attack, so it's got low affinity. Okay. Let me see if there's questions online here. So the affinity is tested against all antigens for a particular B cell. So we have to compare all antigens against the B cell before coming up with the affinity score. You come up with the affinity, so this loop loops over all antigens one at a time. So this this repeat here is kind of repeating the for loop. So this whole thing sort of lives within this for loop here. So for one antigen, you go through all of this, calculate the affinity for that one antigen, alter your population based on that one antigen, and then you go and you do it again for the next antigen. So you do all of this stuff for each antigen, and then you loop the whole thing again, and based on the new population of B cells that come out of this process, you can start the whole process over again. So it may be that, or you can run this once. So you can maybe, it might be you terminate, maybe even only once. So you could do this process just once for all of your antigens, and you may actually get a pretty good match. Or you can repeat this process with the new B population that comes out of this whole mess and try it all over again, hoping that you'll eventually get these kind of multi-objective classifiers that do well at matching a bunch of things. And so, I don't know, for me, again, this does feel like a multi-objective optimization where I would actually think a true multi-objective genetic algorithm might actually be a much better tool for generating this sort of variation for clonal selection. But I think the authors of clonal selection were focused on maintaining a lot of diversity in a population as opposed to finding particular solutions. And I think that's kind of some of the decisions here were about that. Is how do I, in as few sort of runs as possible, generate a diverse set of outputs, memory cells, that can find all of my antigens, and maybe multiple ones would find the same antigen. So I got a little bit more robustness. So it's, I think, the idea there. So this, again, you could run this just once. You could repeat where you've got this B that comes out of the first run, ends up getting inserted at the top again. You could have some sort of termination conditions that make it run a couple of times, or maybe you run it continuously and then you can dump in new attacks periodically, and that might reshape your population as you're jumping in the new attackers. All right, so any other questions about clonal selection? As with the GA, there's different ways you can vary this. I just wanted to give you kind of a feel that when we talk about adaptive immune systems, there's just ways of generating novel, sort of ways of generating, like especially in the negative selection case, it's ways of using these kind of population-based methods to generate novelty that is like way outside of what we might be able to come up with if we did it directly. Clonal selection is a little bit more targeted because you actually have a set of challenges you're trying to match, but it still is a way of using these kind of evolutionary algorithms to, rather than solving a particular problem, it's to generate a novel mixture of solutions that hopefully will have longevity and be a creative way to solve problems that we don't even know how to solve. So, all right, so those are our adaptive immune systems. Those are, I hope you can see, adjacent to genetic algorithms. And so in the last few minutes here, I want to sort of pivot to talk about things that are maybe even more correct or more tied directly to genetic algorithms, but generate, I think, even more creative solutions. So specifically things that are, we're gonna start talking about genetic programming. Genetic programming, another thing that Stephanie Forrest is focusing on here at ASU. All right, so one of the, so I mentioned here that these classifiers, these B cells or these ways to detect attacks, you could imagine that the ways to detect attacks here might not be as simple as choosing a bunch of frequencies from a frequency distribution. It might be that you actually have different algorithms that have different sort of decision trees, like the way that you would detect an attack might actually require some logic. You might need to actually probe this thing that's coming in. You might need to say, what's the state of the world right now? If I'm being attacked right now, not only do I see this particular thing that's coming in, but I also might notice that my system's in a compromised state and I need to sort of probe that. These B cells, it would sure be nice if they were not just static entities, but they might be programs themselves. And so that motivates ourselves to think, how might genetic algorithms be able to write programs themselves? How might you be able to say, all right, genetic algorithm, I don't want you to just find me the exact fuel air mixture I need for this jet engine. I want you to actually write me 3 pages of code that solves for the first 100 digits of pi. Or I want you to here's a program that may or may not have bugs. I want you to find the bugs and automatically patch them. So these are things that we would like the genetic algorithms to do that don't feel like optimization problems exactly. And so the GA has evolved to be able to handle these sorts of things. And so there are a number of different, so back when the GA was being formed, so there's a guy named, in the kind of the history of the GA, there was a guy named John Holland who's largely associated with the genetic algorithm. And his focus was to generate something that was motivated by evolution, but also was possible to be analyzed mathematically. And so the first GA from Holland and so on was more limited in its choices for operators, for example, than you might imagine it could be. So there wasn't necessarily a lot of diversity in the GA because it would be difficult to analyze. And so Holland, not only his contribution wasn't just the genetic algorithm, but it was this mathematical schema for understanding how genetic algorithms work, where he could actually prove for certain optimization functions, the GA wasn't a meta-heuristic, it actually would find the exact solution. So Holland thought that the GAs couldn't just be a way to find good solutions, they could actually be a way to find these solutions, but he had to constrain them to do the mathematical analysis. Other people at the time were less worried about the math. And so if you're interested in that, I recommend to look up some of this stuff and about schema, genetic algorithm schema. These are sort of the mathematical tools that were used to analyze what exactly is happening when things recombine, what exactly happens with mutation, and so on and so forth. Mathematically, how can we model this so we can say exactly what a GA is doing? So that's what kind of Holland's focus was. But there were a bunch of other people coming up with evolutionarily motivated algorithms that were not worried so much about the math. And they allowed the formulations to then stray a little bit into areas that were harder to analyze, but maybe played in spaces that the GA, that made them different from the GA. And so as an example, one of the alternatives to the GA were the so-called evolution strategies. Or the ES. And so evolution strategies are, Holland was an American, evolution strategies were a German work, and you often see them characterized with 2 numbers in parentheses, number of parents and number of offspring. And so the initial version was the 1-1 version, one parent and one offspring, where basically each parent is replaced by, is mutated and replaced by its child if the child is better performing. So this is almost like a trajectory strategy, like a taboo search or something like that, where say each parent mutates and is replaced by child if child outperforms parent. And this was the initial evolution strategy. And then they started to create multi-membered versions. And those multi-membered versions would maybe have random mating, crossover, mutation, of course, and selection. Also mutation, selection. And so here, you might have M parents and L offspring. So you can imagine that this is sort of representing that you have this number of parents, a certain number of them are required for recombination. Recombination happens, there's mutation. Ultimately, you produce these L offspring, which may replace some of the parents. And so these approaches were attempts to kind of add in sort of general characteristics of things that kind of felt like evolutionary algorithms without sort of the worry about the formalisms that Holland wanted for the math. And they ended up having sort of a Brownian walk feel to them. And I'm just putting that out there to show that there were, and I'm putting ES as one of these. This was one of the things going on in parallel with the GA. Now, what we're going to start talking about a little bit more then is another thing that was going on in parallel with the GA was someone who realized that, Holland was focused, how do I solve optimization problems in a way that is mathematically convincing? The Germans and the evolution strategies said, how can we be inspired by the evolutionary evolution, but we're less worried about the mathematical formalism? And so they created a whole framework that's very similar to the GA, but they had their own sort of notation for it. That's kind of what I'm saying for it here. In parallel with both of those was the so-called evolutionary programming movement. Where here the focus was, how do we use evolutionary techniques to write code or to build programs? And so it's very similar to genetic algorithms, but they've given up the sort of string representation of the genome. So very similar to GA, but no string encoding of chromosomes. And consequently, no crossover. So all variation is by mutation. And so I'll go into more details about this next time, but just sort of as a preview, the focus was on building finite state machines. automatically. And so, you know, these are your standard sort of formalisms for what goes on in a computer program. You've got different states. You've got different ways to get out of those states. You've got different ways of getting back to those states and so on. And they form these networks. And you can imagine that you could choose when to add new nodes. You could choose when to add new links, and so on. So these are the things that will happen when you mutate these things. So rather than writing formal code, these strategies, these evolutionary programming strategies, generate abstractions of code that we then know how to, once it's built this network, to translate down into code. So they don't work directly with the code, they work with the states that all computer programs end up implementing inside them. And this was the original way that people came at this. Later on, which we'll go over when we get to genetic programming, they found that there are ways to actually have the genetic algorithms write the code directly. And that's kind of the more modern way that we use these evolutionary strategies to generate programs. And that's where we're going, and that's where we'll start on Thursday. So that's all I've got for you today. If you have any questions, feel free to come up after. Otherwise, I'll see you in a couple days. Questions online? So before I end the room, are there any other questions online or anything, clarifications for me to take care of before I end? If not, that's all I've got for you, and you can feel free to take off. All right, see you all later.